So something I want to make clear is that natural selection does not create variation. Natural selection is an editor or an eraser. It removes phenotypes that don't work in the environment. So one thing to keep in mind is natural selection directly works on phenotypes, because that's how organisms interact with their environment. But of course, when you remove phenotypes, you often then remove genotypes and change allele frequencies, right? Because they're all related. So if natural selection is always whittling away phenotypes that don't work, how do we maintain genetic diversity? Because we know we have to have variation in place, because evolution is a contingency game. Contingency means you're always ready for something new to change, right? You always have variation in place. You have to have variation in place in case the game changes, in case the climate changes, or food sources change, things like that. And how do we maintain that diversity? Well, there's a couple ways. One is neutral variation, meaning we have a lot of genetic diversity that is neutral. We mentioned this earlier in the lecture. This is where there's no real selective advantage to the diversity, but it does increase diversity, and perhaps that trait could become useful at a later date if things change in a certain way. An example of that is human earlobes, whether they're attached or not attached. Having attached or detached earlobes, as far as I know, doesn't mean you're any more likely to have babies and reproduce, right? Or survive. But it is variation that we have. Another thing to keep in mind is because natural selection only works on phenotypes, the expression of genes, and the fact that most organisms are diploid, or some are even more than diploid, some plants, for example. Therefore, a lot of traits are hidden unless they are unless the organism is homozygous for that trait, for example. So, for example, you might have a trait, maybe you're big R, little r, and maybe big R is selected for whatever that trait is, but little r still gets passed on and inherited because it's recessive and it doesn't always cause a phenotype to display. Okay, now an example of another thing that can cause variation to stick around is that sometimes being heterozygous at a particular locus is better or conveys greater fitness than someone who's homozygous. It's called the heterozygote advantage. For example, perhaps being big A, little a is better for you than being big A, or little a, little a. And this is the case for sickle cell, especially for populations living near the equator in areas with lots of malaria. So if you are big A, you don't have any sickle cell alleles. You don't have any sickle cell shaped blood cells. If you're little a, all of your blood cells are sickle shaped. Okay? So if we look at the results of that, so first of all, let's remind ourselves of this. If you have a sickle cell allele, that means that at one position you have an A where there should be a T, and vice versa. That means your hemoglobin has a sickle shape, which means that your red blood cells take on, an emergent property of that is that red blood cells take on this shape here. So hemoglobin's shaped incorrectly, red blood cells take on a sickle shape, is what I wanted to say, and they can actually interlock with each other and block blood vessels, which can lead to cardiovascular problems. Whereas most of the world, individuals have kind of roundish disc-shaped blood cells that don't interlock with each other, right? But why would having this red blood cell shape like this occasionally be an advantage? Well, actually, let me go back. In individuals that live in malaria-rich environments, if they are big A, meaning wild type, meaning normal red blood cells, they can be killed by malaria very easily, or get very sick from malaria. If they have little a, they'll be very sick from sickle cell anemia. Okay, but if you are a heterozygote, big A, little a, your blood cells are typically normal shaped, but when you are infected with malaria, the infected cells kind of go sickle-shaped, and your pancreas can easily filter those out and destroy them. And so it gives you the ability to fight malaria. So you have an advantage over the rest of the population. Now, if you move from Africa to Europe or the United States or wherever, that's no longer an advantage, at least now, because we've gotten rid of malaria for the most part. So it's no longer an advantage because conditions have changed. But that isn't as a great example of the heterozygote advantage. So having big A, little a is an advantage that keeps variation for those two alleles in the gene pool. I just wanted to end with this image here. We see a herd of modern elephants here, and their tusks are pretty impressive. But if we look at elephants from 100 years ago or longer, they had often much bigger tusks. And what we can see here, you can think of as kind of a combination of natural selection, or you might call it artificial selection just because people did it. But people would hunt elephants with big tusks, and they would shoot those elephants. So that meant elephants with big tusks had fewer babies. Therefore, big tusks didn't get passed on in the gene pool as much, so most elephants today grow much shorter tusks because we removed a lot of the big tusk alleles from the gene pool. So variation allows for organisms to change and adapt. And just remember, when I say allows organisms to change and adapt, I mean populations, not individuals. All right, that's enough for this chapter.